Good evening, everybody. I'm removing the mask, but I'm far enough away from you. When I say welcome, you say hello. Welcome.
Hello.
Welcome.
Hello.
Welcome.
Hello.
It's starting to get a little chilly in here. I don't know how we're all feeling. Can we just shake out the hands? While you're doing that, take a look at the people around you, because we are going to be sharing the space together today. And we're going to hear stories, and we're also going to add our own questions, our thoughts, and our feelings to the stories that we hear. So I'm just going to begin. If this were possible, if there was a way, if we could change things, understand and know, our knowledge increasing, our findings grow, together we would reap the benefits that our collective consciousness already knows. Or, if for the hell of it, if for the fun, if for the necessary, if just because, ifs to be answered, ifs to be proved, to be disproved. What is proof? Ifs for the me in you and the you in me. Ifs that are small, ifs that are big, ifs that are insignificant and don't matter at all. Ifs that can change what we think from one thing to the next. Ifs that are infinite and become fact. And become fact. They become fact. Become fact. If he had said, if she had done, if they'd done things differently, if we were to become, if he was right, if she was wrong, ifs were actions, if we were to try, if we were to stop, if that could be undone, 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 if we could do, what ifs matter to you? My name is Amantha Edmead. I'm a storyteller and I'm a drama practitioner, and I'm going to be with you as we go through today. This evening we are going to hear 4 stories which are going to intertwine with our own thoughts, our own questions, our own ideas, to create a story which can only happen tonight with the people that are here. So you've heard my voice, you've been introduced to this festival, and one of the key things about the types of storytelling that I do is that it doesn't just happen with the people supposedly that you see on stage or standing in front of you. It doesn't happen without you, without each and every one of you. So we're going to start with the most important parts or people that make up this space, and that is you. So I want you to take a moment and think about what you were expecting from tonight, or what you are expecting from tonight. And on the last part of my thoughts on ifs, think about what ifs matter to you. So just take a moment for that. And I'm imagining that we've all come in our bubbles. And what I would love you to do is take a moment to turn to the person in your bubble or the person behind you. Say hello, be polite, and also share what you're expecting to get from tonight, and the whole idea of the If Festival and science, and if anything is possible, what kind of ifs are important to you. So go ahead. You can turn to the person you know if you know no one. You can move a little closer. People are wearing masks because of COVID. Just a moment to hear our own voices in this space.
expect to try and do my best to write on the
When you're ready, have any last thoughts, and then we'll come back together. So any last thoughts? Doesn't have to be. That was a very quick, last thoughts were over very quickly. Okay, we shall continue. As has been mentioned today, we are going to hear some thoughts from 4 scientists about their journey and about their work, and these are going to intertwine to make a story. What we want to do today is, as we hear the 4 different segments of our stories today, we're going to take a few minutes after each one of those for you to have any initial thoughts, comments, anything that strikes you, and then once we've told all of the 4 different tales, we're going to open out to questions and answers which may have been stirred from hearing all of the stories together. It may be a burning question. So we're going to go by introducing each of the different scientists, hearing what tale they tell, then having a moment of any feedback that you want to give, anything that strikes, or even just a moment to let that settle, if no words are coming, and then we'll move on to the next one. Yeah? So, sometimes it's good to start with a beginning. And that is what we're going to do. We are going to start with a beginning, where we have a scientist at the beginning of something, sharing something which in many ways only affects a very select few people. But what if we were to think of the I in the few? What if the few were you? What if you were that I who this was happening to? I'd like us to take that thought into our first sharing. Our first sharing. So, I would like to introduce a scientist to you who is going to tell the first section of our tale. Archana, would you like to come up? We have comfy chairs, we have a microphone for you, and you can use whatever you would like. But she can't just come up here in silence because we're all telling this together. So can we have a round of applause, please? You can actually use this one.
Hi. So I'd like you to meet PCDH19. Hi, I'm PCDH19, and this is a tale about me and a girl called Archana and our journey together. So I'm a gene. I live on the X chromosome. Both men and women have two sex chromosomes. Men tend to have X and Y, and women tend to have X and X. So PCDH19 lives on the X chromosome. So we're going to speak from PCDH19's point of view now. So when he or she, okay, let's make him a boy. So when he carries a mutation, he tends to affect little girls in their infancy. So he gets really angry and he sets off these terrible seizures which start at infancy and just go on and on. And they sort of cluster together. It's very difficult. And what happens when he does that is that they don't respond to medication for a very long time. And when they grow into teenagers, their seizures stop, but then the damage has already been done because they're left with deficits, intellectual deficits, behavioral deficits, and so on. So he's got a little secret, and we want to know what he's doing. Because PCDH19 is an X-linked gene, traditionally, he should not be affecting girls this badly. See, because women tend to have 2 copies of the X chromosome, even if 1 copy of their chromosome is damaged or they carry a mutation, they usually tend to have another copy that can compensate for the damaged copy. Men don't have that, they just have 1. Sorry guys. And when this happens, women are usually offered better protection from severe disease on any gene which is on the X-linked chromosome. But when this guy comes into the picture, it all completely changes. So he's carrying a secret that scientists around the world are puzzling to find out. So this is where I come in. I'm Archana. I grew up in India. And ever since I was a kid, I was really fascinated by trying to find out what mysteries were in between the folds of the brain. I always wanted to be a neuroscientist. I was so fascinated by this mysterious organ. So I enrolled for a course in genetic engineering. I did an undergraduate program in genetic engineering, and it wasn't until my last year that I actually became a full-fledged researcher. So it was like letting a kid into Disneyland. That's how excited I was. So I was selected to do an exchange program at Boston. And I'm from India. I'm from a bustling city like Bangalore. So I still remember boarding my flight out to Boston. I was so nervous and I was so excited. I was really nervous because I was going to a different country where I didn't know anyone. They drive on the wrong side of the road. I don't know what vanilla chai lattes are and all of that. But I was really excited because I was going to be in a research environment. I was going to be setting up cool experiments, mixing my own reagents, recording my own results, all without the pressure of a practical exam. So when I went into this research environment, I had the best experience. I got to participate in seminars, which I'm pretty sure I was not supposed to be at. I got to interact with scientists, I shared the lab space with some amazing people, and I got to learn so much. It truly was the best experience, and it cemented my desire to be a neurogeneticist. But then, motivated by that, I applied to do a master's at Imperial College London, and I flew from Boston to Imperial. Imperial was a jolly ride. It was amazing. Until my research project came along. This time, it was a little bit different. So instead of being in a lab, I was in an office in front of a laptop. My project this time involved a lot of statistics and programming. It wasn't exactly my strongest suit at that point of time. And I remember spending long afternoons in front of the computer, just bashing my head because every time I was just editing and re-editing commands on the program line and nothing would actually work. It was intensely frustrating and it would always come back with an error. Every time that you learn something, you're bound to fail a couple of times before it actually works. So that happened a lot. I found out how lonely research could be when it's just between you and your computer, and how hard it could be. I genuinely missed interacting with people in the lab space or, doing all these cool experiments I thought I was going to do. But it was a very important lesson because it did give me time to pause and think if I really wanted to aspire for a career in the sciences. Because both of my experiences are very integral to a life in research. So I realized that I really wanted to do neuroscience at the end of it. But then I hadn't still stumbled on the topic that I wanted to do it on, which is why I chose to wait. And in the meantime, I chose to work in neurosciences. So I came to Oxford, I got a job at NECN, thank you for that. And I spent 2 years as a clinical care coordinator in an epilepsy trial. And it was when I was sitting in an epilepsy masterclass that it truly hit me. I wanted to study PCDH19 epilepsy. I was so overjoyed that I finally found something which was so puzzling to scientists across the world. And I wanted to take it up and, just run with it. So I remember being really excited and going up in front of my laptop again, but this time it was more excitement, I promise. And looking up for scientists all across the world who were working on the same gene that I was interested in, which is this guy, and emailing all of them saying, I'm so excited, can I please come and work with you, and all of that. The response was probably lukewarm. Actually, I lied, it was really bad. It was crushed, it just crashed. There were no responses in the beginning, and then when they did start coming in, they were very discouraging in the sense that we have no funding, it's such a rare disorder, we have no capacity, and all of these are absolutely legitimate reasons in the sciences. So I remember walking into my then supervisor's room on a very bleak November evening, being very discouraged. I walked in, he's a very calm and erudite man. He exudes a sense of calmness, and I remember walking in, taking a deep breath. I said hello, I sat down, and I immediately burst out. Nobody wants to work with me, but this is such an interesting disease. Why does nobody want to work with me? How do I do this? How do I do that? So he gave me a very measured glance, told me to take a deep breath, and told me that there was a researcher in Oxford who was also working on the same gene. I didn't know about this. I emailed the entire world, but I forgot to check my own backyard. But yeah, this tale has a good ending. He helped me email the researcher in question. We exchanged a few more emails, a meeting was set, and 2 years later, I'm now a DPhil student at the Taylor Lab, working on understanding PCD-H19 epilepsy.
Wonderful, Archana, thank you. Thank you for sharing that. I know the temperature is starting to drop in here, but I just want to take a moment for if there are any initial thoughts that come from hearing that part of our tale. Any thoughts or any questions? Or comments?
I want to know what the PCD gene does when it works. Properly.
It's a protocadron gene, so it's involved in calcium binding in the central nervous system.
In what? In the central nervous system?
In the central nervous system, so it's a protocadron gene. It's involved in cell-cell adhesion and calcium binding.
Okay, I see. And so when it goes wrong, that means the signaling between the nerve cells doesn't work properly or something like that?
Yeah, when it goes wrong, it's because of a mechanism called Mosaicism. So women, because they tend to have 2 copies of the X chromosome and one of the copies is damaged, you will have PCD-H19 negative expression, and the other chromosome is activated as well because of X inactivation. Something goes wrong there, and you tend to have PCD-H19 positive cells. So when you have these 2 different groups of cells existing at the tissue level, that's when everything goes wrong. It sort of sets off these seizures, and they're generalized seizures, so they come from all parts of your brain, and then they sort of wean off towards your late teenage years, or early teenage years.
So does it start cascades of electrical activity in the brain or something like that?
It is hypothesized to. So there's still ongoing research, but it is hypothesized to.
Thank you for your questions. Go ahead.
Is it effective in the fetus?
Not that I know of. Every single paper that I've read so far, no. I haven't come across one as yet. Because it arises as a de novo mutation in most cases that I've read of so far. So, but the ones that are inherited can be checked for them. I think they can type for them. So yeah. But mostly not that I know of.
Go ahead.
How rare is it?
So it affects 1 in 10 girls under the age of 5 who already have seizures. So it's quite rare, but in terms of like epilepsy genetics, it's the second most commonly identified cause of seizures in kids.
Any other thoughts at this time?
Have you forgiven the people who didn't take you on around the world yet?
Yeah, I mean, that's part and parcel of the game. I'm not bitter about it. I landed up in a good place. So, yeah.
I work on a gene that has the exact same inheritance pattern.
So we can't hear the questions you're asking.
Sorry, I work on a gene that has the exact same inheritance pattern as this. It's actually really interesting that because can you prove that it's mosaicism? Or is that just a hypothesis?
I think there's a paper out by an Australian lab from the University of Melbourne which has proved that it's mosaicism, at least in PCD-H19. And there have been studies about mosaic males. This is mostly Italian research papers. But yeah, mosaicism is pretty much identified in this condition.
Okay, so my American prof friend is convinced it's dominant, ours is dominant. I kept saying it's mosaicism. It's nice to read that paper.
Any other thoughts before we continue? Okay. Can we have a nice little warm to warm us up round the floor? We are going to continue on our journey. We are now going to hear another story to add to our collective story. This story carries on with the idea of looking at something which is familiar to us, something which we may now have grown to love, to hate. And I think our next researcher is going to share with us ideas that might change our love-hate relationship, that might change what could be called miseducation in some ways. So I would like to ask our next researcher, Yin, to come and join us on the stage, and a big round of applause for him.
Thank you very much, Amantha. And as she says, my story is centered on a carbohydrate, or carb to his friends. Now, carb was born in the leaf of a plant and matured in the seed of a wheat plant. But then it was rudely crushed and eaten by a human while dunking him in some milk. Now, what's even worse is that humans are really mean about carbs, right? Humans just think that carbs just make them fat and give them diabetes. And Carb is really convinced that he does not want to get burned for energy or to get turned into fat and live on someone's midriff forever. And so, as Carb went into the bloodstream through the gut, he was determined to escape this fate. And just as he was swimming through the blood, a really glutinous transporter protein called GLUT, that's really its name, dumped him into a cytoplasm of a cell. And from there, Carb entered the new world of discovery that I'd never seen before, of incredible complexity. The cytoplasm of a cell is like a busy marketplace, filled with biomolecules of every kind, zooming around and rubbing shoulders with proteins of every shape and size. And just then, Carb noticed the massive structures of the cell, like the round nucleus in the center of the cell, with its many spaceport-like pores, and connected to it, this infinite maze-like structure that's called the ER. Next to that is this giant stack of pancakes with these bubbles bubbling on and off each layer to each other layer constantly, as well as going into every other part of the cell, including to the outer membrane where they merge with the membrane itself. And just as Carb was mesmerized by this sight, a bunch of proteins rushing, one sticks of phosphate on him, while the rest try and pull him apart. Carb screams and says, "Just don't break me down!" Because you're a carb, that's what you do, you know, cell needs energy, we need to break it down for energy. He goes, no! And then Carb screams and runs away as fast as he could until he runs through a crevice of the ER where another protein pulls him in. Goes, so here you don't want to become broken down with energy. Come with me, I'll help you. He goes, Carb says, with tears in his eyes, I'm a carb, can I be anything else? He goes, of course you can. Carbs are some of the most important molecules in the cell, and they serve a whole variety of functions. But first, let me change your flavor. So these energetics enzymes won't try and break it down. Because change flavor? What do you mean? Well, actually, in the cell, there are over 50 different flavors of carbs, and only a handful are used for energy. Good. Okay then, change my flavor. So the enzyme changes its flavor and says, right, now we need to activate you so you can partner with a lot of other types of molecules. Wow, that sounds fun, says Carb. So another enzyme joins him to a nucleotide. Carb says, wow, I feel so much energy. Because yeah, now you're ready to partner with a lipid or a protein or even an RNA molecule. Wow, what would you like to do, Carb? Carb said, proteins seem to run this place. I'd like to partner with an important protein. So okay, you need to go down the protein glycosylation pathway. What's that, Carb thought? Well, Carb said goodbye to these friendly proteins and then went off to search for the glycosylation pathway. As it moved through the many crevices of the ER, it came across a bunch of proteins using activated sugars just like him to build a small tree. He goes, what are you guys doing? Oh, we're building a glycan to go down the glycosylation pathway. He goes, oh, I want to do that. Okay. Then they add him to the growing carb tree called a glycan. And as they do that, another protein flips it into the inside of the ER, into the lumen of the ER. And there, a series of proteins in a conveyor belt keep adding more and more and more carbs onto the glycan tree until finally a tree of 14 sugars is made. As soon as it's made, Carb notices a giant upside down snowman made up of lots of different proteins and RNAs. It's called a ribosome, and that's when Carb notices a wriggly little string coming out of the ribosome, wriggling ever longer, wriggling furiously trying to find its shape, failing all the time. And then ribosome looks at this growing string, which now looks like a pile of silly string on the floor, and says, oh, you need a glycosylation. And he shouts to his friend, Ost, come over here. This one needs a carbo support. So Ost trundles along, and this is a complex of 8 different proteins, and says, right mate, okay, who's the little fella that needs some carbo support? He goes, that one, look, it can't even fold properly. Okay, so Ost goes, oh, right, I need to go find a glycan now. And that's when Carb says, ooh, I want to help. He goes, ah, you're perfect. So Ost takes the whole glycan tree and plonks it onto the protein, sticking it to it. And that's when a couple of other special chaperone proteins come along and they grab onto Carb and the glycan tree for purchase and they yank the protein this way and that way and this way and that way, folding it in amongst itself until finally the protein finds a stable structure. Ah, that's better. Right, now what I do? You need to go find your purpose, the ribosome said. Okay, what do I do to do that? It goes, you need to go to the Golgi. There, you can go to every part of the cell. So Carb and his friend go off to the Golgi via a little bubble shuttle. And they merge with the lowest level of the Golgi. Now the Golgi is like a giant train station. Like any train station, it has lots of gates to check what tickets you have. Well, in this train station, the Golgi, all the gates are more proteins. So these protein gates look at Carb and his friend up and down, look at his structure, checking out its carb, goes, right, you need to go to the next layer of the Golgi. But first, I need to trim you, Carb. And so Carb goes, what do you mean? Right, I need to take a few sugars off and then add a few more exotic flavors, kind of bling you up a bit. Okay. And then will you tell me where I need to go? I don't know where you need to go in the end, but you need to go to the next layer. And off they went on another bubble shuttle up to the next layer of the Golgi. And there, we find another gay protein, we trims carb a bit more, make it even more exotic, and then so on and so forth. Layer upon layer upon layer of the Golgi it goes, until finally they get to the top layer of the Golgi, at which time Carb feels exhausted. Please, someone tell me where I need to go. So finally, the final gate protein says, I need to know where you need to go. You need to go to the outer membrane of the cell. What? What do you mean? Isn't that dangerous? I have to go outside the cell. He goes, well, yeah, you do, but why do you think we've been making yours fancy, Carb? He goes, oh, yeah, I was wondering about that. Why am I so bling now with all these exotic flavors of carbs? But well, you know, all these proteins on the outside, you have lots of peptidases and enzymes of all kinds. They will break down proteins whenever they recognize and can take a hold of them. But when you have lots of fancy carbs, they can't get hold of you, so they can't break you down. Ah, okay. So carb and the protein got onto the final shuttle bubble and merged with the outer membrane, where they finally see a lot of other proteins that look just like them. They all have carbs on too. Finally, the protein says, one of you tell me, what are we doing here? What is my purpose? And they go, well, you're a serotonin receptor. And that's when they notice that there's, across a small gap in front of them, there's a membrane to another cell. And out of there, some bubbles emerge, and these molecules come out flooding into it. And one of them gets stuck onto the protein. Mysteriously, a hole opens into it. It goes, what happened? I got a hole in me. And then it sees these salts rushing through sodiums and calcium. And then the cell gets super energetic and activates and depolarizes, sending a nerve signal straight to the rest of the cell. And that's when Carb and his protein partner finally realize how important they are. They are the receptor to the signal that makes us feel happy. And they are essential for us to feel happy. And that makes Carb feel happy. Thank you.
What a journey Carb takes in that. Now, I want us just to take a moment, and before I was asking if you had any questions or any thoughts, I still want us to have those thoughts with us, but one of the first things I'm going to ask you to think about is, in that story, which is very different from telling just your straight personal experience of how you came to do what you're doing, in that story, what surprised you and what was new? Just take a moment. Go ahead.
I think what surprised me was, I thought it was complex, but quite how complex it is and how that came across in your story with all of the different layers, and the fact that what we eat seems to have such a direct impact on, but in such a way that it is broken down in many ways and then builds up in many other ways and sort of has to go on all of this journey. That was, I didn't expect it to be that complex.
Thank you.
I just want to elaborate on that. So the protein glycosylation pathway actually occupies some 3% of our genome, so it's something like 300 genes of our genome is dedicated to this process. And so that's why there are literally hundreds of these enzymes that modify the carbohydrates and move them along for this particular process.
Anyone else got anything that surprised from hearing that story? Oh, go ahead.
Sorry, I was just going to say, how do the gatekeeper proteins know what to do? How do they decide how to make them bleed?
That's actually a really good question, and I'm not sure we really understand, but we think it's to do with the structure of the protein as well as the carb. So not all proteins, so when people look at the glycosylation patterns of proteins across the cell, it's very distinctive for each different protein. So for example, the glycans on lipids and things like that. And so a lot of these proteins have very specific sugars on them, and only on specific proteins. So these enzymes must recognize very specific structures, and we don't know how they really recognize these things. And equally, a different type of glycosylation you mentioned today called O-glycosylation, we still can't predict which proteins get glycosylated. So it's still a massive mystery.
Go ahead.
Sorry, me again. It sounds like you followed the sort of pathway of a molecule all the way from ingestion to its use in the brain. It's got to be hundreds of chemicals like that to do. What sort of percentage of these pathways have been sort of studied and mapped out, and how many more need to be understood before we sort of understand brain chemistry to that sort of level?
It's a very good question, thank you very much. It's very difficult to say exact percentages, and it will be different for every different organism.
Right.
So I was kind of talking in a simplified and direct story where the carbohydrate goes straight to your gut into a neurochemical. I'm ignoring the fact that there is, a blood-brain barrier that it has to go through first, and then it probably then gets into your brain. And even inside, there are actually a lot of different chemicals. They modify the sugars as it goes through. It's very complicated, and frankly, in humans, we have no idea because we can't experiment on humans.
Right.
So we have an idea in simple organisms like yeast, or even in cultured cells from people, usually morselized cell lines of some sort. But in a person, in terms of what proportion of the sugar, I can tell you a rough percentage of the sugar. About 20% of the sugar you eat is burned for fuel by the brain. So this is part of the reason why people are physically weak, because our brain uses so much energy. But in terms of what proportion of the sugars in the brain goes into protein glycosylation, I don't think anyone really knows, I'm afraid.
Go ahead.
Thanks for the story. Very interesting way to describe the process. Is there any study that tries to look at how different diets could affect these production of receptors? You gave the example of serotonin, but maybe a large spectrum.
Thank you very much. It's a very difficult study to do. Certainly there are people who have studied those who have, let's say, diabetes, who have a lot of sugar circulating into their system, and see how that then affects the glycosylation patterns in, let's say, their serum. So materials that are more easily accessible to a scientist. Obviously, we can't take someone's brain and work it out, but there are definite differences. And actually, there is a type of glycosylation that is transient and can come off and on the protein. And recent studies have shown a lot of the effects of diabetes could be because of different levels of glycosylation.
Thank you. And for this moment, any last question or thought before we continue with our stories? I think we shall continue. We have a warm. Thank you for your story. We are going to continue with our next episode of our stories. Now, this is a story that in some ways combines what happens within how our realities can be then changed by our experiences. So we're going to continue looking at the body, looking inside the body, and looking at all the different impacts that that can have with our next story. So I would like to invite up to the stage to share our next story, Adam. Big warm
Okay, so this is almost a real story. Deep within a young girl's body, in the darkness and silence of flesh, something goes wrong in one egg within an ovary. The egg divides, but this parthenogenesis is not miracle, but more nightmare. It expands into a monstrous mass of fibrous sinews, matted hair, abortive teeth, and jumbled nerves, a teratoma. However, the young girl's body is not defenceless against this monster within. Serried ranks of white blood cells scan and sniff around the mass, waiting to taste something that shouts, "Not self," so they can begin their attack. After years of waiting, one white blood cell scents something foreign and dives into the monster, expanding into an army of clones and sending out the call to arms to its colleagues. This cascade of killing exposes one white blood cell to a brain protein that should not be there. In response, some of the attacking cells start to pump out antibodies to destroy the monster's brain. One of the more intrepid amongst these brain killers heads off into the girl's body to hunt for any monsters that may be lurking elsewhere. Now we zoom out to see the whole girl, Sally, no longer a girl, but rather someone on the cusp of adulthood. Sally is putting her bags in her mother's car ready for university, full of excitement for what is to come. Her mother drops her off at university and they bid a tearful farewell at the door of her new room. But just as her mother is leaving, Sally hears a faint bell ringing somewhere in the distance. She strains to hear it more clearly, listening with all her being, but then it is silent. She snaps back to the world in time to hear it, in time to see a look of concern on her mother's face, but summons a sad smile, sufficient to reassure her mother for the homeward journey. Now alone, she hears the bell chiming again, this time accompanied by a low, sibilant whisper, chanting incomprehensible words and the faint scent of sulphuric smoke. Rewinding to just before the drive to university and zooming once more back into the world inside Sally, the intrepid white blood cell has caught the scent of the monstrous brain protein, a trail leading from nodes in her neck into the liquid blackness of the fluid around Sally's brain. Hovering below her hippocampi, the seat of her memories and the rich vein of experience that makes Sally Sally, the cell detects a vast amount of the brain protein peppered across a gently buzzing network of nerves and synapses. It sends out a distress call to other white blood cells waiting in the teratoma, summoning an exponentially increasing army to wage a war of mistaken identity against Sally's brain. The first antibodies are launched towards Sally's hippocampi, streaking forward like torpedoes, and the elegantly orchestrated buzzing of Sally's nerves adopts a disordered, frenetic crackle of misdirected electricity. Back in her college room, Sally greets her new roommate, Kathy. She is barely able to hear what Kathy says over the hypnotic tolling of the great brass bell, echoing from an indistinct distance, which then falls abruptly silent. To break the ice, she makes her roommate a coffee. As they sit down to drink it, Sally realises she has forgotten her roommate's name and, more worryingly, can no longer recall the word for what they are drinking. Swiftly taken to the college nurse, she is reassured that anxiety around the first day at university can manifest in many funny ways. Back in their room again, She watches as the far wall of her room dissolves into flames, and clawed hands reach up to replace her roommate's eyes with orbs of hellfire. She tries to shout out a warning, but her words fail. Her right arm begins to jerk uncontrollably, and darkness rises up to steal Sally away. The war against the girl's hippocampi is almost over. Antibodies have coated all of the brain protein. The collateral damage of this pitched battle is apparent everywhere. The gentle hum of ordered nerve activity has been replaced by arcs of iridescent discharges, flooding out of the hippocampi to rob her body of movement and speech, before casting her identity adrift on a sea of misfiring electrical activity. Snatches of awareness only now. The roommate's face, looming over her with eyes of burning crimson and a smile twisted with teeth within teeth. A sanitised room with tubes flooding the girl's body with poison, and a woman with kind eyes asking her questions, unaware of the winged horror perched on her shoulder. Frozen in place, without free will, a statue of wax to be moulded by a puppeteer. Shadows within and without contort her limbs into impossible positions. Now being drawn into a fire-ringed tunnel to hell, surrounded on all sides by the banging of devilish drums and the wailing of lost souls. A woman almost familiar, weeping while the room dissolves into kaleidoscopic shapes. The pull of the shattered windows, the cool, soothing blackness of death beckons her through the aperture, speckled with her own blood. Cruel hands drag her back from blissful extinction as the almost familiar woman splits into 1,000 wailing banshees. A gloved hand of the supernatural anaesthetist, smothering and suffocating her while whispering words of false reassurance into unhearing ears. Knives cutting into her stomach to birth a monstrous horror, howling as it leaves her body. But now, the tide of the battle turns. Steroids pumped into her veins soothe the anger of the attacking cells throughout her body. A sterile knife carefully removes the teratoma, along with its twisted, abnormal nerves. And thereafter, the torrent of white blood cell reinforcements flooding the brain dries up. Gradually, the cells that started the attack on the girl's hippocampi drift away to rest. Cautiously, the brain protein begins to re-emerge from its concealment within nerves, and the howl of misdirected electricity settles to the former organised hum of synaptic activity. Gradually, Sally begins to coalesce from the fractured fragments of herself. One month after the removal of her teratoma, she looks at the woman standing in the hospital room and says, "I love you, Mum." She reconstructs a narrative of herself through such small victories. After a month spent enduring the boredom of rehabilitation, she is released back into the care of her family. The terrible process that stole her away now has a name: NMDA receptor antibody encephalitis, which we now know to be an uncommon but increasingly recognised cause of neuropsychiatric problems, seen in around 1 in 200,000 of the population. Its one kindness to her is that the last thing she remembers is being dropped at university by her mother. She survived the war within and regained her name and self as Sally.
Thank you. So we continued with our journey inside and out. I want us to just share anything that's come up from that for us. Any questions, any thoughts, or any observations?
Thank you for the story. I was wondering, is it this condition correctly diagnosed? Like what you described seems to be tricky to recognize.
So do you mean is it recognised, tested for, and then treated?
Yeah, like is it nowadays addressed correct?
Yeah, so I suppose we don't know how many are missed for sure, but it has a very specific order of symptoms that often come on. So nowadays we're looking for it so we can recognise it and test for it. Historically, if you look at some of the old studies of people before this protein was recognised, actually of the people who had this based on historical testing of bloods, about 90 to 95% of them either died or had terrible disability. So that's completely transformed now.
And what is the epoch we're talking about? Like, is something that is changing in the last 50 or 10 years? Like,
When it was recognised, you mean?
yeah.
So over the last sort of decade or so.
Thank you.
Can I ask, is this something that people have heard about before, or is this a new revelation? I see shaking of heads, so not heard of before from people who are here. Okay. Go ahead.
It just reminds me of Brain on Fire. It's a book about NMDA and encephalitis as well, and it's very similar. The story was very nice.
Thank you. And a film as well, I think.
Yeah,
isn't it?
yeah,
Yes.
So did everyone hear that reference, that it reminds you it's called Brain? Go ahead.
Brain on Fire, yes. So you can check out. There's a movie with I've forgotten the name of the lead actress actually, but yeah, if you just Google it, you'll be able to find it.
If it helps, I just remember his Brooklyn Better Jack's girlfriend.
Thank you.
So it's both a book and a film.
It is.
Go ahead.
How accurate is the description of how the girl responds in the room at university and what happens to her at that point? How do you know?
So I've definitely taken poetic license, but actually based on, so we did a study a while ago looking at all the published literature and what the patients report, and also within Prof. Irani's group with whom I work, they've done a lot of interviewing of patients when they recover. Now, actually they have almost no memory of what happened during these times, but we know that there's a great obsession with death, with all sorts of funny visual imagery that goes on, and they have the waxiness, sort of the feeling like a marionette. That's what they behave like actually. So it's the most bizarre thing. If you see them lying in bed and you reposition their arms, they will remain in that position stuck until very gradually they relax back to normal again.
It was a very powerful description.
Thank you.
I may have missed the point, but did you say anything about what triggered this process?
Well, so we don't fully understand that, but the fact that when you look at this condition in general, about a third of patients will have this unusual teratoma, so the mass sitting in their pelvis, and they'll have had that for a long time probably. It's a very slowly growing thing. But within that, there are elements of central nervous system, so nerves, supporting cells and so forth. And at some point, for reasons that we don't fully understand, that triggers off an immune response against the nerves there. But of course, there's no real difference between the nerves in the teratoma and the nerves in these patients' brains. So there appears to be an attack that starts here, then proceeds into the brain. But once you actually get rid of the original material that was making this sensitization or immunization, actually the attack, if you do it quickly, seems to die away. It's really remarkable. They go back to, they're often almost back to normal by the end of it.
I'm still left with that thought that it's only in the last 10 years that this has become known.
It's really transformative actually, yeah. Yeah, it's been amazing.
Okay. Have we got any last questions or thoughts at this moment? Then I think it's a big warm. We are now moving on to our fourth element of story. In this, we again are meeting a scientist and the work and the scientist behind the work. So we're almost coming full circle with the journey of it. Never, in some ways, the human cannot be separated from the work that we do. And I'd like to welcome to the stage, Amantha Edmead.
Can you hear me? Yes. So, I wanted to be a neuroscientist for a long time. I love thinking about how we make decisions and love reading everything I could find about the brain. Like most people, my idea of what a scientist was came from movies, science textbooks, short biographies of Nobel Prize winners. I imagined someone very curious about a problem, that would stay awake at night trying to solve it, and would eventually crack it. Of course, I understood that there may be some variability in the timing that it took, and that maybe there were some complexities, but I believed that at its core, this was it. So I wanted to study the brain. Some scientists study the brain. So I wanted to be a scientist. So I went to university, I did internships, and eventually I entered the PhD program. It was a dream to be studying exactly what I wanted and surrounded by so many clever, passionate scientists that, at least from what I could see, fit this idea. So I started my work, and for the first couple of years, everything went well. I was lucky, my experiments worked, I got exciting results. I was over the moon. I remember thinking to myself, okay, maybe I can do this. But then, things changed. I started an ambitious project, working on something very new that no one had worked on before. Initially, it was really exciting, figuring things out for the first time. But then I hit a big roadblock, a big technical challenge that, unless I managed to overcome, would prevent me from answering the question I was after. At the beginning, I was full of energy looking for solutions. And every time an attempt failed, I doubled down my efforts. I remember thinking, a good scientist would figure this out. If I were a good scientist, I would figure this out. And so I worked day and night, on weekdays and weekends, always telling myself, you can rest once you solve this. But this went on for months after months after months. And the more I focused on work and on solving this problem and left everything else in my life fall behind, the more crucial it seemed. You are a failure, the thought would pop in my mind. Now to get back to work, running away from that thought. So I kept at it, and I kept at it, until the point in which I was too exhausted to even care about the answer on the other end. Eventually, it got so bad that I couldn't get out of bed. I was diagnosed with major depression disorder. I tried many medications. I went to psychotherapy. And eventually, after many months, I started feeling better. Slowly, I started to feel some joy again and interest in the things around me. And it took a while, but eventually, I even recovered the passion for neuroscience. So I went back to the lab to finish my PhD. I doubted that I was going to solve the problem I had been working on before, but I was lucky enough that the work from my initial years had gone well, and I had already published it, so I knew that at least I could graduate. At that point, I was pretty sure that I wouldn't make it as a scientist. Not only because I didn't crack the big problem I was working on, and I saw that as a big failure, but now mostly because I was the only person that I knew in my entire institute that had had a depression. But exactly because I didn't feel I had much to lose, I stopped hiding my struggles. So when people asked me what had happened over those months and what led to it, I told them. And the most surprising thing happened. People from all ranks started sharing with me what they considered their failures in science and all the mental struggles that went along with it. Importantly, just like me, they thought they were the only ones. I was struck. These were amazing scientists, yet they felt this way. And knowing about their struggles didn't make me respect them any less. In fact, I admire them even more. This realization brought waves of relief. Maybe these ideas that I had about what a good scientist was were just wrong. I became sure of it months later when at a conference I was talking with a senior scientist, one of my big science idols. We had met a few months before, and he knew what I had been working on. So he told me they were looking for someone with my experience and asked me whether I would be interested in joining their center after I finished my PhD. I was filled with excitement. But pretty quickly, the excitement turned into doubt. But, I said hesitantly, I haven't solved the problem yet. He turned to me and with a kind laugh said, "Come on, you're a PhD student. No one expects you to have it all figured out. This is a really hard problem. Even if you don't crack it within your PhD, you've learned an awful lot. You know better than anyone what doesn't work and why. And this is really important, and this is what we want. We'll figure out the rest together." Another wave of relief hit me. I had thought that maybe I could get a job with my earlier work. But this last project, I had considered a complete failure. And so I relaxed a little bit, and we discussed some of the details of how things would work out if I joined their center. I felt happy, but this other thought kept coming back. You need to tell them about the depression. You cannot afford to hide it. My body tensed up. I looked down at the pavement, trying to avoid seeing his reaction, and I said nervously, I had a pretty serious episode of depression during my PhD. My supervisors were very supportive and they let me take some time off, and I'm well now, but I can't guarantee it won't happen again. As the words came out of my mouth, I felt naked, completely exposed. And what followed were probably just a couple of seconds, but they felt like a lifetime. Eventually, I ventured to look up, and instead of a frown, I was met with a kind smile. He said, "I'm sure we'll all be very supportive." I was so relieved. I smiled, and I thought to myself, you should really scrap that idea of what a good scientist is. For so many years, I tried so hard to be like this scientist that I had in my head. Too hard, to my breaking point. And it turns out I didn't have to do it at all. So now, it's time to make sure that others don't fall into the same trap. Thank you.
Now, have we got any immediate responses to what we have just heard? There don't have to be. Go ahead, please.
Why do you think it is that we as humans treat imbalances in the brain with a different attitude to say imbalances in if you break a leg or some other physical attribute? Why do we look at it differently?
It's a very good question. So I think part of it is that the physical part normally is visible to some extent, and even the part that was not visible, now we have tests, blood tests and scans to look at it. Whereas mental illness has been much, much harder. It seems to be so complicated, and we're still so very much in the beginning of understanding and having markers to say, look, this person has depression, for example, just looking at a test. We still rely mostly on self-reports and on criteria of, okay, this person has this symptom and has that. And so I think that also makes it, even experiencing one, and I know that's also one of the symptoms of depression, but they were people report, and I certainly felt as well, is that you're constantly doubting your own symptoms. You're thinking like, oh, everything is so effortful, and retrospectively you can see it was really because of the disease, but at the time you start thinking, oh, I'm being lazy, or why am I not doing? So there's a whole, I think there's an aspect of being an invisible disability at the time, but then also, historically, years of also not speaking about it, and everyone feeling that it's a measure of their inadequacy rather than something that everyone experiences.
Thank you. Go ahead.
If you went back and had your time again, would you tackle the same hard problem?
No. Even though I'm still working on it here, but I think it was too much. It was too hard, and I think for a PhD student, and especially in a context in which I was the only one doing it, and because my project was a collaboration between different labs, none of my supervisors actually had the expertise that I had eventually, and so it was very hard to also feel confident about and ask for help. And so I think it was more a team project rather than one PhD student at that time, and so yes, I think it can certainly happen at all levels, especially as a PhD student when you're trying to figure out whether you can be a scientist, and I think it's very hard. So I would probably not have come to that.
One of the things, sorry.
Is there a difference between a brain of a person who becomes depressed and someone who doesn't? There's a picture of a brain up there.
So we certainly cannot tell just looking at a brain like that. We also don't necessarily know whether even a group of depressed individuals are an homogeneous group, or actually different groups within something that we call depression. But yeah, we're not yet able to look at a person's brain and say this person is or was depressed. So I guess that's where we're trying to go, but we're still far away from that point.
Go ahead.
I'm going to stare at the elephant in the room here. What is that massive problem that you have to tackle?
So it was to we use MRI in humans we do a lot of other research in animal models that we cannot do in humans. And the project was to make it possible to do MRI in animal models, so then you could more directly translate what we know from animals to humans. So it is still very important, and I hope we'll figure it out, but yeah, it turned out to be incredibly hard.
Go ahead.
Has anyone asked you to explain to supervisors or people in charge of students about what the situation for someone in your position, how it could be so serious? Have you been called upon to help some of your superiors in a way, explaining about how it came about?
So you mean called by someone to say maybe this other student is going through this and whether I could give some advice, or?
Yes, or in general, just to staff who are in charge of PhD students about the problems and how serious they could be.
Yeah, so I think there is gladly starting to be a big movement, and certainly, for example, in our department, I'm part of a mental health working group that is a big step, because the leadership has acknowledged that this is a really important aspect for students, also for staff, and that, you know, it's across all professions, but also there's things specific about our profession in terms of an uncertainty of funding, and so what this group is trying to do is, one, provide training for supervisors to detect but also know what to do about this, and also in general something that I think academia needs a lot, which is a change in culture, a change in talking about this, in acknowledging and understanding that this is not weakness, this is not failure. This is something that actually, if we look at the stats, most people must have gone through it, even though we don't know. So yeah, something that helped me a lot, and when the science idol in my story that also told me was, he started telling me about all these people that he knew that, scientists that I had long admired, that had struggled currently with mental health issues, and were still great scientists and were great mentors, even if they had to take some time off once in a while. And that helped me so much, because then you don't feel, okay, maybe the fact that I had depression means I'm not cut out to be a scientist. It just shows you that we're all humans and we all go through these things, and it doesn't mean anything about your quality as a scientist. And as you say, I hope, if anything else, that at least this will make me more sympathetic and careful with my future students.
Go ahead.
So, have you gained any insights into how to distinguish whether a problem is currently solvable or not currently solvable?
This is what we wished we knew, because even that's something that, you know, supervisors sometimes maybe try to hide because it would scare you too much in the beginning of a PhD. But even supervisors don't know what project will be safe or not. I think in general, if it's something following up on something else, maybe that doesn't require as much development, and maybe it's a little bit safer. But even there, things can still not work out. In general, yeah, I think in my project, for example, it was a high gain, high risk, clearly. And so maybe I wouldn't have chosen it for a PhD student. And indeed, I think I was the main one pushing for it, probably because naively I didn't understand that.
But yeah, it's very hard. I think you never know what's going to work. But I think that perhaps the most important skill is to then, when something is not working, decide when it's time to drop it and move, and then maybe, okay, this is not working, let's move to something else. But incredibly tough call to make.
Thank you. I'd like to take a moment to open this out to all of the scientists who have shared their stories with us tonight. We literally have just under 10 minutes left, and we've got one exercise which we want you to go away with, but before then, is there any questions or any thoughts that you have for any of our scientists here, or anything that you feel you'd like to say in this space? Oh, I see a little slow hand going up. Go ahead.
It just struck me how everyone who said, we don't know very much, we don't know very much, we don't know very much, and there's still so much to know. And I suppose I thought that was, I don't know what I thought. I think it is incredible that we still have so much to learn, yet we already know so much as well. But I don't know. That was just thought.
No,
it's not very well thought of, but
it was clear that we don't know, and yet we know so much, and there's so much still. Even though we're looking at completely different people's experiences and different disciplines. So thank you. Any other thoughts or specific questions to one of our scientists here? Oh, go ahead.
I just wanted to say thank you to each of them, because it seems from the stories you've shared with us that to advance the aspects of science that you're each dedicating yourselves to, it's taken exactly that, dedication. It's taken a large portion of your life, or a portion anyway, that clearly was very unpleasant. You experienced the rejection, as nearly everyone on the planet does, but you experienced lots of rejection, and I'm sure your colleagues have had to dedicate large portions of themselves to this. And I appreciate your choosing to do it, but there are positive outcomes that other people benefit from as a result. So it was just a thank you, really, for doing your job.
Thank you. One of the questions that we had in the beginning of this is the IF Festival. It is about science, it is about ideas, it is about starting a dialogue with people. To find out what you're interested in, and also anybody feel free to jump in. Not only what you're interested in, but how in some ways people can work together, or we can demystify some of the ideas behind science and scientists. And yeah, so one of our questions to you to go away with is there anything, what would you want from the IF Festival? What is your IF, as it were? And if, for example, we have here people who want to be scientists, already are scientists in their own ways, what would you need from us? So, what is your if? Is what we would love you to put down on these little index cards that you have on your seats. And any questions or thoughts that you have in that regard about demystifying all the connections that can be built going forward. So I'll give you a moment to think about that. And I don't know if we have pens or if everyone already has their own pen in these days of COVID. Pens or pencils to note down your thoughts. And they can be completely anonymous. So it's totally up to you if you want to name. As you're doing that, I want to echo the thanks that were offered to each one of our scientists who have come up today, and shared their work and shared their experiences with us and shared themselves. It is a real honour and a real privilege to hear your stories. Very excited to see where your journeys will take you and us. And one of the things as we leave today, as we continue our journeys, this festival is happening once a year, but we'd love there to be a connection throughout the year. So it's not just we appear once a year. So we're here, work is happening, and to feel that you are able to reach out and connect, if that's something that you want to. And from our scientists, would you like any last words also? Or have you laid all your words out and shared your stories?
First, just to thank everyone for coming with you.
Yeah, thank
yes,
you
I
all
was
for
going to
coming.
say.
Yeah, thanks for the F Festival. I think it's fantastic. You know, I think I first got involved a few years ago before COVID, and it's just brilliant. I love the openness of it. So thank you to the organisers for giving us the platform to speak.
I would also like to add thank you to Jacqueline and Nikki for facilitating this for us.
Yeah.
And for listening to me practice.
And also Amantha
Yeah.
for turning this into a coherent story.
Yeah.
It's an honour to hear them. So I feel that we have come to the end of our session today. If anyone, just before we go, because there's something about adding our voices to this space and to this recording, from where we are there any ifs that are still in your mind that are important to you? That was our initial question as well. Would anybody like to voice any of those? Okay. I want to say thank you all for coming. Thank you for joining in, thank you for your questions, thank you for your energy, thank you for your presence, and thank you for your time tonight. And we hope to see you at other events, and I don't know if anyone would like to say anything else before we close. And if not, travel safely, thank you for coming. Please fill out any of the evaluations that you can, and travel safely. Thank you.